then. Todd Smith's professor in biology at Acadia and has done lots of wonderful research over the years, including some of friends of the Blomidon Naturalists as his research mentees, I guess they're mentees, whatever. So Todd's going to talk about the secret.
Okay, let's see if this works first. Can you hear me?
Yeah.
Excellent. Okay, well, thank you very much. I have to say this is my first time here at Blomidon Naturalist Society Nature Talk. Every time there's one of these, I have a teaching lab at Monday evenings, and by the time that's done around 9:30, I'm sure this is all over. So I kind of had to make this one, obviously, because I'm speaking, and also school's finally done for the year, so the night was free. So again, thank you very much. It's great to be here. And I want to talk a little bit today about malaria parasites of frogs and snakes. And you might not know that malaria parasites in frogs and snakes are literally about 2 kilometers from here down the Oak Avenue Marsh. Actually, that might even be about 1 kilometer from here. We actually have malaria parasites in frogs and in snakes very close to where we are right here. It's not human malaria, as we're going to see in the talk, but it is a type of malaria-like parasite that these animals get. I'm just going to take out my phone here to record this so that I don't run on past too long into your evening. There we go. Okay, good. Okay, so I've been interested in frogs and snakes since I was a very young boy. My father was a birder, and he took me out family time so he could bird watch and not have to be at home. So he took me with him. And at 3 years old, I really didn't know how to use binoculars or anything. So he would look at the birds and look up, and it was really too small to do that. So I just kind of started looking down, and so I started seeing a lot of things like frogs and snakes, and was always too slow to catch them. But they kind of caught my eye, and as I got older, I think I was about 7 years old when I caught my first frog, and about 8 years old when I caught my first snake, when I was finally fast enough to do that. And now I get to do it as a living, which is I couldn't ask for anything more there. I've been working on frogs and snakes and their malaria parasites for about 33 years now, since I started my PhD in Toronto in 1992. Okay, let's see if this will go. Okay, there we go. So, secret lives of malaria parasites. Some major themes for today. I want to start off with what type of species of amphibians and reptiles are found in Nova Scotia. You might have seen them. We've already talked about a couple of species already. And then I'll talk about what challenges are encountered by frogs and snakes in Nova Scotia and worldwide. That's in the title. There's a lot of challenges that they face just naturally that they've evolved with over millions of years, but also some human-induced changes that they now have to deal with as well. I also want to talk about the idea of a parasite. You probably have a pretty good idea of what a parasite is, but I'll give some definitions just in case you're a little bit unclear on what we humans try to categorize as a parasite, because they are a major focus of what I'll be talking about. And then I also will talk about malaria parasites and what are they doing in frogs and snakes, excuse me, and also in mosquitoes in Canada. Now, I couldn't talk about frog and snake malaria without mentioning mosquitoes. There was a talk by Laura Ferguson a couple of months ago on ticks. Today we get to talk a little bit about mosquitoes. And then also, if we have time, I'll talk about how climate change may impact malaria parasites, not just in frogs and snakes, but also in humans and in other animals as well. And in case you're wondering what I do in the summer, professors don't have the summer off. We do research. And so here is me. This is taken about, I guess, about 6-7 years ago. And this is me in the middle, a little bit more hair. This is Claire Bullock, who's now in her medical residency, and Amy Prescott, who's now a medical resident, or a physiologist rather. And we're collecting frogs in a pond on the South Mountain. I think it's Lockhart Mountain Road. Phyllis Harvey owns this land in this pond, and it's one of our go-to sites for collecting green frogs. And some of the research that I'll talk about happened there. And here is in the animal care facility where we house the frogs and snakes. Here is a little cave where they're living in one of the terraria, and there's three heads of maritime garter snakes sticking their head out of there. We'll talk a lot about those as we go through the talk. Okay, so let's talk a little bit about vertebrates, because I mentioned these animals, vertebrates, animals with a backbone of bone or cartilage. And you're probably familiar with most of the groups that make up the vertebrates. We have the jawless fishes like lampreys and hagfishes, the cartilaginous fishes like sharks and rays. And if you've ever been to Guy's talks, you probably know all about rays. And then there are the two groups of bony fishes, ray-finned and the lobe-finned fishes. And then we have amphibians. Animals like frogs and salamanders, they've got a thin and moist skin that is thin enough they can actually breathe through, but they have to keep it moist, so they're restricted to either wet areas, moist areas, or actually the water itself. And their eggs aren't very well protected. They're just covered in like a jelly-like layer. Reptiles, things like turtles, lizards, snakes, and crocodilians, they've got their skin covered in scales, and they have a more protected amniotic egg that often has a shell, although there are some snakes and lizards that give live birth as well. And then, of course, we have birds and we have mammals rounding out the other groups. And I just thought I would put in the non-reptile and amphibians. And I thought I would put my favorite species of fish, which is the brook trout, my favorite species of bird, which is the black-throated blue warbler. I'm not a very good photographer, so that's taken right off Wikipedia. And then I had to think, what's my favorite mammal? And I had to think hard. I think it's probably the red fox, and it came up in the talk today. So that timing is quite good. Okay, so that's what vertebrates are. And the two groups I'll be talking mostly about are amphibians and reptiles, of course. And I thought I was going to go through the web and try to find some pictures. And then I thought the good thing to do would be to take John Gillen's book of Amphibians and Reptiles of Nova Scotia that he wrote in 1984. And I have a copy with me if you'd like to look at it. Beautifully illustrated by Fred Scott, who passed a couple of years ago. He was our museum curator for quite a while at Acadia in the Department of Biology. And when I was looking at John Gillen, I never met him. I realized he just passed 2 weeks ago. And this is almost like a tribute to that book and some of the artwork from Fred Scott. So amphibians in Nova Scotia, here are the salamanders. Sarah mentioned the yellow-spotted salamander. They're moving around right now. Be careful driving around on rainy nights because they're heading to their ponds to lay eggs. The blue-spotted salamanders, more in northern Nova Scotia. We don't have them very commonly in Kings County. Red-spotted newts have this aquatic stage, and then they've got a terrestrial stage as well. And so these are often found in ponds, and you can find these in forests. Four-toed salamanders, a little bit local, not very common. I've only seen one in Nova Scotia. And then redback salamanders are one of our most common salamanders. And neither of these species of salamanders has lungs. They breathe through their skin. They've lost their lungs over evolutionary time because they can just breathe, take oxygen from the environment right over their skin. In terms of amphibians, frogs and toads, they're calling now, and they've got a fairly set sequence in Nova Scotia of how they call. The wood frogs are usually first, and then a couple days later, the spring peepers will start. The wood frogs kind of sound like a little bit of a quacking sound, almost like almost ducks with a sore throat. Spring peepers are calling now, come in all different different colors. The toads will follow soon. They'll be calling in another couple of weeks or so, a long trill. And then we've got green frogs and leopard frogs coming next. You'll get some, these are the green frogs here. Sometimes they come in a blue color, if you've ever seen a blue frog. Leopard frogs are not as common as they are in other parts of Canada, but there's lots here. Green frog is probably our most common frog. The mink frogs are a little bit later. These are fairly local. They like big lakes with lily pads. And bullfrogs are the opposite. They like really warm, kind of stagnant ponds, and they're our largest species of frog. Pickerel frog's a little bit local, looks almost like a leopard frog, but is usually brown instead of green. In terms of reptiles, we've got some turtle species, 4 of which are freshwater and on land. And then some are visitors from the ocean, sometimes come to our shores. We've got the snapping turtle here, we're found throughout Nova Scotia. The wood turtle, fairly local. Most people keep it their distribution a secret because they are poached in Nova Scotia. Blanding's turtles are only about 500 or so left in the Kejimkujik area. And then the painted turtle, very widespread, but nevertheless still threatened. It takes them a long time to start breeding. And then the ridley, the loggerhead, and the leatherback have been observed offshore and sometimes stranded on beaches. In terms of our snake species, we'll be talking lots about the eastern garter snake in Ontario, maritime garter snake in Nova Scotia. They're part of a species called the common garter snake that's found all over North America. And we get this fairly non-striped garter snake. Most garter snakes are striped in the rest of North America, but ours are kind of spotted. You might get the odd striped one, and sometimes they come in an all black phase as well. Red-bellied snakes and the northern ribbon snake, this is pretty much only found in Southwest Nova. These three species all give birth to live young as an adaptation to northern climates. They don't have to worry about laying eggs or anything like that and keeping those above cool temperatures. But the ringneck snake and the smooth green snake, these are egg-laying species, and you may have seen these throughout the province. Okay, so that's kind of introduction of the amphibians and reptiles in Nova Scotia. There are lots of species around the world, and our reptiles and amphibians and the world's reptiles and amphibians are suffering from a number of challenges. Again, some of them have been around since these organisms have evolved, and so these are the ones that they've adapted to. Nevertheless, they are challenges. Challenges encountered by frogs and snakes. Let's talk about predation, competition, infectious diseases, things that are naturally occurring, don't necessarily have anything directly to do with us. There are lots of predators of frogs and snakes. They're not the largest vertebrates in the world. They are food for a lot of organisms. Lots of mammals, foxes and raccoons will eat frogs and snakes. Birds, herons love frogs and snakes. This is not a common Nova Scotian species, but this is the gray heron that Sarah found in Miners Marsh about, I guess, 5 years ago now. So this is, I'm no photographer, but I did actually take that picture there, and it's not too bad, I guess. Very photogenic bird, just kind of sat there for everyone. So birds, herons love to eat frogs, and they'll take snakes as well. Snakes eat frogs, and so that's a predator for frogs. Snapping turtles will eat frogs, and they'll eat tadpoles. Bullfrogs will eat anything that they can stuff in their mouth, which includes frogs and small snakes. Large fishes will eat frogs and will eat small snakes. Aquatic insects like giant water bugs can catch those newts and suck them dry. And crayfish, we've got a couple of invasive species of crayfish now in Nova Scotia. They can eat tadpoles, and apparently they can eat small snakes as well. So they're coming. Great. So that's predators. Competition amongst either the same species or amongst closely related species. They're competing for the same food. Frogs and snakes are, or frog species are, and then snake species are. Both frogs and snakes are predators, so they themselves are, they're competing for food, and also space. That pond that I showed you, Phyllis Harvey's pond, let me just go back a couple slides here. So that pond there is only about maybe 20 meters across. There's about 60 or so green frogs in there, and they each have their own little bit of turf that the males defend against all other males. So you wouldn't think of frogs as having to compete for space, but they do. So that is something that competition amongst different frogs, or even amongst the same species of frog, for food and space is a thing. In terms of infectious diseases, ranavirus. I had a whole year of frog work wiped out by ranavirus. It's a type of skin and almost like a flesh-eating disease that causes these lesions, lethal lesions on their skin. I lost another year of research to chytridiomycosis. caused by this thing, which we just call BD, mercifully shortened to BD, the Trachochytridium dendrobatidis. It is a type of skin disorder, a fungus that lives under the skin and causes the skin of the frogs to slough off, and then they can't breathe properly anymore, and that's lethal. The frog mutating fluke, it sounds like it's a type of worm. It sounds horrible, and that's exactly what it is. Riberoia ondrate. The larval worms live inside the developing limbs of the tadpole as they start to sprout their legs, and it causes them to sprout extra legs. And this frog is not going to be able to get away from herons, get away from the other birds. Birds are the actual final host for this fluke parasite, and so they get these larval worms when they eat these sick frogs. The frog has basically been disabled by the parasite. So it literally is a frog mutating fluke. Even if they don't get eaten, they rarely live past about 3 or 4 months. And we did have a frog that had this disease that we caught this year just on that pond on the South Mountain. And here's a new disease, a snake fungal disease, SFD. It's in Ontario. It's in Quebec, and it's slowly moving here. There was a survey done by a researcher in New Brunswick who found about 25 snakes in Nova Scotia. Couldn't find it yet, but it probably is going to be coming. Another fungal disease that lives under the skin of these snakes and ends up causing them to get secondarily infected by bacteria, and this can be lethal to them. Okay, so moving on then to challenges encountered by frogs and snakes, human activities. These are things that these animals have not evolved with. They're more recent in terms of their evolutionary time, maybe only the last 200 or 300 years, or maybe even sooner than that. And so this is not a comprehensive list. This just gives you an idea of some of the challenges. But habitat degradation is a huge one. And certainly all of you are aware of that. Degrading, losing habitat for forestry, for building houses, fragmenting habitats so that the nice stretches of forest or marshland are broken up by highways. Even a highway through a marsh can fragment a habitat so that frogs no longer can get from one side to the other, and they have to go over the road, often with disastrous consequences. So that's a huge one. That's a big one in Nova Scotia. Chemical contamination, things like pesticides. We're in an agricultural area. Pesticides are widespread. Fertilizers widespread. Wastewater, industrial pollutants. All of these affect frogs and snakes, and especially frogs, because that very thin skin they have, many of these pollutants can go right through their skin and can poison them that way. So chemical contamination is a major problem. Invasive species, we know invasive species can be problems. We know that starlings take nest holes from our native birds, but it is also a problem even in the frog world. American bullfrogs have been introduced all over the world because apparently, never had frogs' legs, but apparently their legs are delicious, and so because this frog basically can eat anything that can stuff in its mouth. This frog has caused the rapid decline of a number of different frog species around the world, including many of the frog species in British Columbia since the bullfrogs has been introduced there. Consumption and collection. Frogs' legs, especially in Europe and in the southern United States, a delicacy. So they've declined in many parts of the world because they're not farmed. Some of them are farmed, but others are collected from the wild. And the pet trade. This is a picture I just found off the web. This is terrible. Lizards and snakes that have been put into these little containers that are going to be shipped all over the world for the pet trade. Snakes in particular worldwide suffer declines in numbers because of the pet trade. And then climate change, we'll talk about this at the end of the talk. Long-term alterations to temperature and precipitation have effects on frogs and toads, frogs, toads, snakes, in terms of things like drought and elevated temperatures. We'll talk a little bit more about that in a few slides. Okay, so I promised you that I'd tell you what a parasite was. And again, you probably have a fairly good idea of what a parasite is defined as. But let's talk about this because it is kind of important to understand what these parasites do in frogs and snakes and in mosquitoes, as we're going to see. So we can humans love to define things. We love to put things in categories. We love to count species and things like that. So this is a definition that is, for the most part, covers most parasites. An organism or infectious agent, and if you're wondering, what the heck is that? Viruses are not really organisms, but they certainly are infectious agents. So an organism is living, viruses, kind of halfway there, infectious agents that lives on or in another organism, and we call that organism a host. So parasites infect or infest hosts. And they often cause a measurable decrease in what we call fitness. And this doesn't just sort of mean, oh yeah, I haven't really been running for 3 or 4 weeks, my fitness is down. This is something that is some sort of change in your life. You may not be able to grow as fast. You may not feel as well. You may not be able to reproduce because you don't feel well. Many of your resources are going into fighting off a parasite. So parasites often cause a decrease in fitness. This is an example of a hookworm right here, only about 1 centimeter long, but 30 or 40 or 50 of these inside your intestine, especially if you're a child growing, can take so much blood from your intestine that these children can end up growing much, much more slowly. And they can even take so much blood that they can actually cause starvation and death. So that is definitely a measurable decrease in fitness caused by little 1 centimeter worms in the gut. And then parasites often cause harm to their host, things like diarrhea or coughing. And those have evolved to help the parasite spread through an environment. And we don't really have to go too far to think about something like norovirus, which is a terrible intestinal condition. It's not lethal, but those 24 hours while you have Norwalk virus or norovirus, and you lose like 7 pounds of water because you're throwing up and you're having diarrhea. Well, those two actions, throwing up and diarrhea, that gets these virus particles into the water, into the air, and they can then spread to the environment into new hosts. Okay, so that gives you an idea of what parasites are. What about the biodiversity of parasites? There's all kinds of different organisms that we can call parasites. Almost all viruses, with very few exceptions, are parasites. I think you might have heard of that one, and there's what it looks like in all its terrible glory. We just talked about norovirus as well. These are definitely very common, widespread viruses. Influenza is another virus, of course. HIV, the human immunodeficiency virus, is a terrible virus. And then ranavirus, I just talked about that, is one, or sorry, that's one that, talked about that one already. I changed the order of these slides just before I got here. Now I'm mixed up. The ranavirus in frogs is another type of virus. Doesn't affect us at all, but certainly can cause all kinds of problems with frogs. Bacteria. Not all bacteria. In fact, very few species of bacteria are parasites, but the ones that are, well, you've heard of the plague. That's not up here, but that, of course, changed human history, killed about 1/3 of the world's population. Salmonella is a type of food poisoning bacteria. Borrelia, Laura Ferguson talked about this a couple months ago. That's the Lyme disease causing bacterium. So that is another type of parasite. And chlamydia, a sexually transmitted parasite. I won't go into the details of what that does to you. So those, again, not all bacteria are parasites. Only a few are, but again, the ones that are, certainly they get a lot of notoriety. Fungi. If you have athlete's foot, that's trichophyton. Get that from your local change room or swimming pool. So that is a type of fungus that lives under the skin and causes incredible itching. Chytridiomycosis, again, I've talked about this. It infects frog species, directly related to the extinction of about 3 dozen of frog species around the world. Didn't just harm the frogs that in my lab that one year, but it's done all kinds of damage to different populations and species. And then there's that snake fungal disease, which hasn't caused any extinction of snakes yet, but it'll be interesting to see what happens as this spreads around the world. Protozoa, I'm going to talk about malaria parasites of humans, that's Plasmodium. And Giardia, if you've ever not filtered water when you've gone camping and days later you have very explosive diarrhea, you might have Giardia. That's a type of protozoa, and these are single-celled microbes that have a nucleus. Bacteria are single-celled microbes that don't have a nucleus, so protozoa tend to be larger and more complex, and we'll talk about those a lot today. And then we've got lots of animals that are parasites. Again, not all animals, but quite a few different groups. We talked about that fluke that lives in frog legs and causes their legs, well, extra legs, the frog mutating fluke. That's a type of worm. Tapeworms, you may have heard about those, can get to 25 meters. That's 80 feet for those people who know the imperial system inside humans. So, your digestive system's only 10 meters long. You've got a 25-foot tapeworm in there. Nematodes can range from anything from little tiny hookworms that are about 1 centimeter long to 30-centimeter roundworms that can come out of your mouth or nose at any time. Ticks, if you were here for Laura Ferguson's talk, I'm not going to go into those. You heard about ticks. Mites are also, some mites are parasites. Here is a head louse on some hair. This, again, is not my picture, but thank you, Wikipedia. And fleas are also animals that are considered as parasites. And then mosquitoes as well. We're going to talk about how mosquitoes aren't necessarily true parasites, but they certainly can carry parasites. And I'll mention that talk about more about those in the next slide. Okay, so mosquitoes. Family Calicidae, in case you're wondering about what family they're in. They're a family of true flies, and true flies are a group of insects that have 2 wings. Almost all other groups of insects have 4 wings. True flies have 2 wings. And so mosquitoes, there's about 3,700 species of those worldwide. 48 species have been found in maritime Canada. And so that's quite a few different species. And they can be day-feeding ones, night-feeding ones. There's all sorts of species. The females, they're not really parasites because they don't really reduce your fitness unless there's 1 million of them taking a little bit of blood. So we call them micropredators. They basically are taking a little bit of your resources. That's food for them, but they're not going to cause you to die unless they give you some fatal disease. So we call those micropredators. Females, they need to suck vertebrate blood, so blood of those vertebrate animals that I listed. They need that to acquire nutrients for the egg development. Males just feed on nectar. They don't need blood. They just feed on nectar. That gives them the energy to go and mate with females. So there's a female, and she's just laid some eggs on the surface of the water. The eggs hatch into what are called larvae, kind of like the caterpillar stage of a mosquito, but they're aquatic. They live in the water, and they breathe through little tubes at their abdomen, at their tail end, and they take actual air from the surface from the surface of the water, and they get the air that way. Then they turn into a pupa, which is kind of like a cocoon, and they also have to breathe through a little air tube right there. And then after about 2 days, the pupa turns into a mosquito, which lands on the surface of the water, takes off, mates, and then the females will lay eggs again. So interestingly, they're the deadliest animals on the planet. They contribute to the deaths of nearly 1 million people per year, not because they take blood and cause you to lose all your blood, but because many of them can transmit some terrible parasites like malaria and worms, other different diseases. Lots of different viruses are transmitted by mosquitoes. So I've got a fun fact here. Humans kill only 431,000 people per year, either killing each other or wars or hitting each other with cars and whatnot. Mosquitoes are double that in terms of number of deaths. So it's something in case you're wondering. Okay. So malaria parasites. These are carried by mosquitoes, and that's why this kind of segues nicely into talking about malaria parasites. We're getting closer to the parasites of frogs and snakes. So they're protozoa, single-celled organisms that have a nucleus that move around and that can actually ingest things, actually eat things. So they're not animals because they're single-celled, but they're the ancestors of animals. And malaria parasites are protozoa in this called phylum Apicomplexa here. I just feel like I have to say that because I think for the talk title or the introduction that was put out with the email said that I work on apicomplexan parasites. What are those? Well, malaria parasites are one of them. So malaria parasites, they live inside blood cells of species of all groups of vertebrate animals. Here is some human red blood cells. There's those little yellow circles here. This is a scanning electron micrograph that has been artificially colored. But here are the little parasites destroying the red blood cell, and now they're coming out. They're going to invade a new red blood cell, and after 48 hours, they're going to multiply, turn into something like this. The whole red blood cell is going to be full of these parasites, and then that blood cell will explode, and the parasites will infect the next red blood cell. Very quickly, you can start losing red blood cells when you get malaria. So they live inside blood cells of all the groups of vertebrate animals, including frogs and snakes and humans, as I'll talk about. Then they develop in the body of an invertebrate animal, like a mosquito. So invertebrates simply are animals that don't have a backbone. So mosquitoes and leeches are blood-feeding invertebrates that can carry malaria parasites. So the parasites will develop in the body of the invertebrate when it feeds on that blood of the vertebrate. And then they're transmitted to vertebrates when that invertebrate bites. So if a mosquito bites or a leech bites and they're infected, they could then pass on malaria. But there's some species, including the ones that I work on, my students and I work on, they're actually passed when the invertebrate is ingested by the vertebrate. So the frog has to eat the mosquito as opposed to get bitten by the mosquito. And you might not think of that as a big difference, but actually it is a fairly large difference in the different groups. So we'll talk about malaria parasites of humans first. You probably have heard of the disease malaria. Human malaria is caused by, again, these protozoan blood parasites. They're part of the genus Plasmodium. That's the actual malaria parasite group. I'm kind of cheating a little bit when I say malaria parasites of frogs and snakes. They're in a different genus. They're not truly malaria parasites, but close enough. It got a lot of you here, so I guess it worked. They're transmitted by the bite of mosquitoes, a certain type of mosquito in the genus Anopheles. We do have a couple of species of Anopheles in Nova Scotia. We do have a malaria vector here that if malaria ever came here, we would have a mosquito to transmit it around. So let's hope it doesn't come here. Used to be in Ontario, but it's not there anymore. We'll talk about the distribution in just a second. So parasites will multiply in and destroy red blood cells in that manner that I talked about in the previous slide. This is a little small here, but you can see these parasites, the mosquito bites, and they go into the liver for a little bit. And then they come out and they invade a red blood cell, which you need to transport your oxygen around your body. And then they grow, they use up all the protein in the red blood cell. They divide it into about 30 little babies that all burst out of the red blood cell, and then they infect a new red blood cell. And this can happen, this can go on for weeks and months until you run out of red blood cells. You can actually die from anemia, which is not having enough iron in your blood. So severe cases of malaria can be lethal. You can have this anemia that I talked about, or they can get lodged in your brain and cause a coma and death. That's called cerebral malaria. Or they can end up in your lungs and cause fluid to go into your lungs. Then you can't breathe anymore and you get a pneumonia and you can die that way. So malaria kills 263 million people per year, mostly children, which is very sad. And it infects that many people per year and kills almost 600,000 people per year. These are very recent numbers. And just to give you an idea of where it mostly is found, the tropics is where it's mostly found, and it's still a major problem in these areas. We're just now working on not me, but people are working on a vaccine, finally trying to make a dent in some of these numbers. Okay, so malaria parasites of frogs and snakes then. So they're not they're not caused by the genus Plasmodium. They're caused by the genus Hepatozoan, which is a different group of organisms. They look a little bit different. They have a different type of life cycle that I'll go into a great depths about. You can't go to a parasitology talk without a life cycle. And I'm going to give you two. I've already kind of given you the malaria one, but the others are going to fit a whole screen. Okay, so this is a garter snake blood smear. So what we've done is taken a little bit of blood from a garter snake and spread it over a slide, and then you stain it up with special dyes, and then you can look at the red blood cells. They've got little nuclei in their red blood cells. That's kind of what those little circles are. And if you're wondering what's special about this one, it's got a little sausage as a guest in there. That little sausage is a single-celled protozoan parasite called a hepatozoan. And we call that stage the gamete. And it lives inside red blood cells, but unlike malaria, it doesn't destroy them. It doesn't multiply and destroy the blood cell. It just kind of hangs out in there waiting for a mosquito to bite. It doesn't cause the same sort of damage at all. So mosquitoes will go and bite a frog or a snake. They'll ingest these gametes as they eat the blood, when they feed. And then the parasites are going to start developing. They're going to start multiplying inside the mosquito, and they develop these big balls of parasites, what we call an oocyst. It's not a oocyst, although you would be, I can see why you'd want to say that. It's actually oocyst. And they get quite large, or even larger than a mosquito egg. You can see them with your naked eye inside the body cavity of the mosquito. Or in some species, they get into the kidneys of the mosquito. We call those Malpighian tubules. There's not going to be a quiz on this later or anything, but you might want to know the name of a mosquito kidney. And they get lodged in these kidneys here, and then they sit there, and they sit and they wait. They don't do anything. They just kind of hide out in the mosquito, and they wait for a frog to eat an infected mosquito, and then the frog gets the parasite that way. And if you're wondering how do snakes get their parasites back, that's a little bit more complicated, and I'll explain that in a couple minutes. Okay, so here's the first life cycle. Ta-da! I drew that. I am a terrible artist. But somehow I drew this, and I don't think it's too bad, really. It almost looks like, it could be in a scientific paper, and in fact it was. Anyway, so this is the life cycle of hepatozoan species of frogs. It only has two hosts, a mosquito and a frog. And if you think, well, that's kind of complicated, it's going to get even more complicated in a minute. And this is the stage. There's supposed to be a red blood cell here with a little sausage-shaped gamete in it. And then the mosquito is going to take blood, and then the parasites are going to develop inside the Malpighian tubules, the kidneys right here. Don't worry about all these stages, but eventually there's going to be this large multicellular stage, and they're packed with all these little parasites. Each one of those there is a little parasite. And then when a frog eats the infected mosquito, not when the mosquito bites, if it bites, that doesn't do anything to the frog. The frog has to eat the mosquito, and then it's going to get parasites first in its liver, and then the parasites are going to multiply there, come out of the liver, hide out in red blood cells, but they don't do any damage to the red blood cells. Do a little bit of liver damage, but not too much. And we've been able to find them in green frogs, in bullfrogs, wood frogs, and leopard frogs. So there's 4 different species we've found these in over the 30 or so years since we've found this life cycle. And so Culex teratans is the type of mosquito that feeds on frogs, and it doesn't feed on humans. It only feeds on frogs and sometimes snakes. It is a frog-specialized mosquito. In fact, it finds frogs by listening for their calls. So it actually uses phonotaxis, the fancy word for saying listening for a frog, and then it goes and bites it. And there's a frog here. That little bit of blood is left over from a mosquito that bit it, and there's another mosquito filling up on the frog. But it has to be careful because frogs like to eat mosquitoes too. That's how they get the parasite back. So they try to go back here where the frog's not looking. But there's been more than one occasion where I've watched, this is inside a little cage right here. We put the mosquitoes and the frog in. The frog go, ooh, food, and we'll eat them. You have to stuff the frog full of crickets first. It doesn't want to eat the mosquitoes. Otherwise, you'll lose all your research animals. So there we go. Okay, so in terms of where these are found, so I did my PhD in Ontario and worked on frogs and snake malaria there for 5 years. And two species that we found in Ontario, Hepatozoan climate, Hepatozoan catesbianae, they're in Ontario. And the second year I was here, first year that I had students that went out into the field and we all collected frogs, we took their blood, and I was really nervous. It's like, what if the parasite was here in Nova Scotia? Wouldn't have to go back to Ontario to work on these things. And the third frog had the parasite, and it was fantastic. So I mean, that's your frog here. So Nova Scotia's got these species as well. And this is a study that was done by Bryant Boulian, an honors student back in 2007 with Roger Evans, who retired from our department, and me. And he went around all over Nova Scotia, couldn't make it quite to Cape Breton, and found both species of the parasite all through here. So both species are quite common. And he collected green frogs and he sampled those and found that 88% of them were infected with the hepatozoan species. We call that the prevalence, the percentage of frogs infected with a parasite. But wood frogs, he sampled those and only 16% of those were infected. And I kind of thought, that's a bit odd. Wood frogs eat mosquitoes. Maybe there's something about the wood frog the parasite doesn't like, or maybe it's something else. And I'll get back to that in just a second. And in case you're wondering what the difference is, Hepatozoan climate really does a number on the nucleus of the red blood cell. It's all kind of chopped up into little pieces there. We don't know if that harms the cell or not. And this is Hepatozoan catesbianae, which doesn't seem to do very much to the nucleus, just kind of pushes it over to the side a little bit. Okay, so I'll get into the whole thing why we don't think wood frogs get parasites in a second. But I want to tell you about frogs that freeze. So 7 frog species around North America, including 2 that are in Nova Scotia, they partially freeze their body tissues. And by partially, I mean like 65% of their body tissues freeze to survive sub-zero temperatures during hibernation. This is a picture of a wood frog that is frozen. It's about as hard as a hockey puck. 65% of its body is frozen. Not inside the cells, that would be lethal, but pretty much everywhere outside of cells, all the rest of the water in the body is frozen. Here is a distribution of the wood frog, and it's kind of good that they can freeze because sometimes they're unable to get away from frozen temperatures. Most frog species, like the green frog that I'll talk about a little bit more, bullfrogs, they kind of bury themselves in the mud of the unfrozen mud of ponds. If the pond freezes all the way down, the frogs die. If you're wondering about toads, they just burrow down into the dirt below the soil, below the frost line, so they don't freeze. But wood frogs and spring peepers here, these are 2 species of frogs in Nova Scotia. They can't get away from the frozen temperatures. They can't burrow, so they just freeze. And they can go down to -6 under leaf litter, which is, doesn't usually get much colder than that. And they freeze pretty much solid. So how it happens is that ice forms around their organs first. Water starts to flow out of the cells, which shrink a little bit. And then really syrupy, gooey cryoprotectants, which don't freeze until well below 0. Things like high concentrations of glucose, so almost like a syrupy solution, or glycerol, which is a type of slimy solution. These now flow into cells, and then even if the temperature goes below 0, they're so sugary that they don't freeze, almost like syrup doesn't freeze, or salty concentrations of salt water don't freeze at 0. So basically these cryoprotectants flow into cells and protect them. The cells don't freeze, and then the frog comes back, almost literally back to life once the temperature warms up again. Okay, so getting back to our question, why are wood frogs not commonly infected with hepatozoan species? And the person that did a lot of work on these frozen frogs, he gave a talk at Acadia. His name's Ken Story. Gave a talk at Acadia during our seminar series and was talking about how wood frogs freeze. And then suddenly I went, wait a minute, maybe that's why wood frogs don't have these parasites. Maybe freezing kills the parasites. And so I set out to do that the next year. And here's what we did. Well, first of all, surprise, and I was surprised, freezing does not kill hepatozoan parasites, but something interesting happened that was almost as interesting. So what we did is we fed mosquitoes infected with hepatozoan species to 22 wood frogs, 11 of which were the control group. They stayed unfrozen. And then the unlucky 11 frogs that were chilled at 4 degrees for a month. You have to get them used to being frozen. That's kind of like fall for them, or autumn. And then freeze them at -3 degrees for 2 days. This is what they look like when they were frozen. Imagine just kind of getting along, you kind of half burrowed in the soil. And it's like, oh, it's getting cold in here. And then you freeze. And so there are 5 frogs here. You can see 1, 2, 3. There's a couple more in here. This was a picture that when we took a photo of the frogs, we took them out of the incubator they were in, and they were frozen. And we could, we touched them, and they were frozen. And then 2 days later, when we took them out of the freezing conditions, they were all fine. You can even see the little hole that this one was in. There's where all the other ones were, hopping around. It's like, we're hungry now. Can you give us food? And they were all of the frogs were fine. They literally were frozen, and they came back to life. So then we took their blood both before and after the freezing, or before they were just maintained at normal temperatures, before and after. Members of the control group, the unfrozen group, lost 98% of their parasites after 30 days. So wood frogs have a very strong immune response to hepatozoan species. They're probably just not good hosts for that parasite. So, okay, well, that was interesting, kind of, but the members of the frozen group only lost 75% of their parasites after 30 days. So the freezing does not kill parasites, because 98% here died unfrozen. In fact, cooling and freezing, we think, we don't have proof of this, we think it slowed the immune response of the frog to the hepatozoan species. By cooling them and then freezing them, it actually slowed down immunity, and the parasites took advantage of that and lived in an environment that otherwise, if they were at a normal temperature, would have killed them. So freezing doesn't kill hepatozoan parasites. It, in fact, slows down their being cleared by the immune response. Okay, I think I might need a plug, because I'm seeing a power light here. So I do have one, if it's easier for me to get it. Yeah. Okay. Glad I brought this with me. Yeah. No. Okay, should be. Yeah, it has to go to this side. Maybe, can this maybe sit here? No, maybe not. Might be okay. We don't need that one anyway, right? No. Yeah, okay. All right, I think we're back. Okay, so effects of hepatozoan species on frogs and mosquitoes then. Hepatozoan species do not seem to have observable effects on growth, body condition, or reproduction in frogs, which is kind of disappointing. Number of studies have been done. It doesn't seem to hurt the frog very much at all. A little bit of damage to their liver, but not much at all. So if you're wondering if hepatozoan species are going to be the next thing to potentially harm frogs, and it doesn't seem like that's the case. Then again, we also can't ask them, how are you feeling? So it might do something to them, might reduce their ability to want to reproduce, but it doesn't seem to cause any body condition changes. However, heavy infections of hepatozoan species can overwhelm the immune system of mosquitoes, interestingly enough, that feed on the heavily infected frog. They can punch holes in the midgut wall of the mosquito. And this is not my picture, but this is just showing parasites going through the gut wall of a mosquito. This is a picture that we took. This is transmission electron microscopy right here. So this thing here is a midgut of a mosquito, of a control mosquito, and it's got some blood in it, but there's no parasites there, and the gut actually looks nice and thick and healthy. This is the gut of a mosquito that fed on a heavily infected frog. After 2 days, its gut was almost completely punctured there. That's not supposed to look like that. Ultimately, this kills the mosquito. So heavy infections of hepatozoan species can harm the mosquitoes, even though it doesn't seem to do any damage to the frog. So that was quite interesting. That was research of Kiva and Amy a few years ago. Okay, so we're ready for our second life cycle now. Since we talked about frogs and their parasites, here's a life cycle of a hepatozoan species of snakes. And this took me 2 years to figure out when I did my PhD, mainly because snakes don't eat mosquitoes. So the mosquito part was relatively easy. Both two species, Culex pipiens and Culex teratans, and they will feed on snakes. Here's a northern water snake from Ontario. Here's a maritime garter snake from Nova Scotia. They'll feed on these snakes, pick up the parasites. They develop in the body cavity now inside fat body cells, not in the kidneys like the frog parasite did. And then they form these enormous oocysts right here, about, again, almost like half a millimeter wide, and that you can easily see inside the mosquito. And these oocysts are full of parasites that would infect a snake somehow, but snakes don't eat mosquitoes. At least water snakes and garter snakes don't. So we looked for some sort of in-between. What eats a mosquito but gets eaten by a snake? Well, let's try frogs. And sure enough, the parasites developed in the frog, but didn't go into the frog blood. That's what the frog parasites do, the frog hepatozoan, but just sits there in the liver and waits for up to 5 years to be eaten by a snake. And then when the snake eats the frog, and I won't get into details of how we did that, then you get 2 rounds of development, basically this multiplication of parasites, and then they invade the red blood cells and the life cycle is complete. So it took a while to figure this out because we did try to feed mosquitoes to snakes directly and nothing happened. But once we got the frog in there, it did eventually work. It was a great day. It took, I think, 20 months, but it was a good day. Okay, so the host and geographic range of hepatozoan species of snakes. We talked about the frog parasite. Hepatozoan Cypodon was a new species that I discovered in Ontario in the water snakes and then eventually in garter snakes. And that was actually my first paper way, way back more than 30 years ago. Didn't seem like that long, but time does fly, in case you didn't notice. Northern water snakes, they are a very good host for the parasite. 18 of 33 samples, so 55%. Water snakes love to eat frogs. Garter snakes, they like to eat frogs, but they are quite happy eating worms and other things. Only 7% of those over the 5 years of sampling in Ontario did I find that they were infected. And 21% of northern ribbon snakes, they also do like frogs more than some other food. These were all found in Ontario. So locations with a circle are where I found this hepatozoan Cypodon, and the locations, the letters without, I didn't find any. So I always had to go back to Ontario to get material to work on hepatozoan and snakes. I went a couple times in 2009 and 2019. My family remembers when I had the snakes in cages when I came back from Ontario. And then in 2021, I found I was actually looking for a rare lark sparrow that Jake Walker had found. And as I was looking up, when I should have been looking down, there was a garter snake there. So I'll collect that. Why not? My permit's still good for another month. Took it back. It had hepatozoan in it. First time in Nova Scotia, 1,500 kilometers away from where we've last found hepatozoan Cypodon, and found it again in 2023 in Aikens Marsh down near Hansport. And this parasite, this is one of my honor students is working on this right now, and it looks a lot different than hepatozoan Cypodon. Those oocysts aren't as massive; they're about half the volume, so that seems different. We might actually have a new species, but we have to send it away for genetic characterization to see if it actually is. So about 9% of maritime garter snakes that I've sampled over the years seem to have this, but it might actually end up being a new species. Okay, so what does it do to snakes and mosquitoes? There's actually been a study, a really comprehensive study on hepatozoan species that infected snakes at really, really high numbers. Their blood cells were I think about half their blood cells were filled with parasites. Didn't seem to affect their growth rate, their body condition. their fitness in any way. They didn't check reproduction. It's like, oh, a hepatozoan of snakes doesn't seem to do anything to snakes either. You know, all these ideas of, oh, look, snake and frog, the hepatozoan's like an emerging disease. It might be with climate change, but right now doesn't seem to do very much to the frog or the snake. However, hepatozoan does do damage to the mosquito again. So this is the hepatozoan Cypodon. This is actually the master's research of Laura Ferguson. Again, if you were at her talk a couple months ago, she was actually my master's student back in, finished in 2012. And there she is there. And I think I know that guy as well. This is from some sort of whitewater rafting, the tidal bore rafting that they did. That's a group picture they had of the lab. Anyway, so for her, part of her master's, she found that infections of hepatozoan Cypodon in Culex pipiens, so that mosquito, significantly reduce the number of mosquito eggs, especially the second time they lay eggs, so the second time they feed on blood. Here, and as Soren said, don't put any graphs in. Nobody want to see any graphs, but I feel this is such a great study that I do want to put the graphs in. There are these really, really large oocysts here that take up a lot of room in the body cavity of the mosquito. If you don't have an infection, there's about 100 eggs per female. If the parasites are inside the body cavity, there's only an average of about 20 or so. And in fact, if you look at this graph here, here's the number of eggs on this axis. Here's the number of oocysts. The more oocysts there are, the fewer eggs there are. So more oocysts equal fewer eggs. In fact, if you get above about 60 of these big oocysts, there's no eggs at all. The female mosquito has been completely, I don't want to say sterilized, but her fecundity, her egg production has gone down to 0. And considering Culex pipiens is also the #1 vector for West Nile virus, people are interested in this outside. So yes, hepatozoan of frogs and snakes doesn't seem to cause much damage to them, but certainly does cause damage to the mosquitoes. Okay, so to wrap up then, I do want to talk a little bit about climate change and malaria parasites. Talk about humans and birds first, because there we are actually seeing some major changes with climate change and malaria parasites. The range of human malaria parasites is expanding northward, southward as climate gets warmer, going up mountains that never used to have malaria because now the air temperature is getting warmer, higher up. So malaria parasite range of humans is expanding because of the changing patterns of the mosquitoes. So they develop quicker when there's warmer temperatures. If there's more precipitation, the mosquitoes will increase. This is causing an expansion of malaria in most parts of the range. It's slowly creeping. Some parts are getting drier and they're actually losing malaria because the mosquitoes don't like the dry conditions. They need it. They obviously need water for their larvae. Range of malaria parasites of birds in Hawaii. If you're wondering, why am I talking about that? It is also expanding due to temperature and rainfall. Hawaii's mountains are getting warmer and warmer, and the mosquitoes that are living in the lowlands and the low, warm wetlands are now starting to move up. And in case you don't know this, and this is something that's one of the great environmental tragedies, avian malaria was introduced inadvertently with infected mosquitoes on ships as early as 1826. So infected mosquitoes with bird malaria, doesn't affect us, bird malaria, and the species, all the songbirds in Hawaii had no immunity at all to it. And malaria has contributed primarily, there's also habitat destruction as well, but primarily to the extinction of half of the 50 species of indigenous songbirds, and many of those other half are in critical danger. This is the EEV right here, which is still, it's not common, it's rare, but it's still around. It's only threatened. So this one you can still see. This one's not so lucky. This is from eBird, where the person who saw or heard the last one ever in 1987 wrote basically saying, I think I've heard the last one, could never find it. And eventually this species has now been extinct for about 40 years or so. So all because of avian malaria, and the birds are getting pushed higher and higher into the mountains because they still haven't developed resistance to this malaria. So climate change definitely impacting malaria parasites of birds. In terms of frogs and snakes, well, We don't know. It's first of all difficult to assess effects of malaria parasites on the health of frogs and snakes. Again, we can't ask them how they're feeling. A lot of the tests to see if their metabolism has changed because of the parasites, that's difficult to do. There's lots of variability between different frogs or snakes. So it's difficult to assess this in the first place. It's even more difficult to do it in the face of climate change. However, we can kind of speculate. So drought conditions may alter aquatic habitats and stress frogs, make those areas, those ponds smaller sooner. In the last 5 years, there's been a couple instances where the ponds that we collect frogs have actually dried up totally. That's definitely going to stress a frog if it has to find a new place to live. Increased temperatures will accelerate the development of mosquitoes and the development of parasites, which means there'll be a greater chance of parasites coming into contact with frogs or snakes. And this one's interesting. Will changing winter temperatures affect parasites that affect hibernating or freezing frogs? Our winters aren't necessarily getting warmer. They are on average, but the cold snaps we're getting seem to be worse. Those cold snaps might be too cold for some of the frogs that can only go down to -6. It might actually be more challenging for them to even survive. Okay, so thank you. I think I might have gone a little bit over time there, but I do want to just acknowledge a few people. Tanya Morse-House and Don Miner, they're the animal care technicians that help me take care of the frogs. My students and I do a lot of the frog and snake care, but they're the ones that make sure all the water is in good shape, and they'll take care of the animals when we're away. Phyllis Harvey and her pond, which again has been a great field site. The Nova Scotia Department of Natural Resources and Renewables. This changes so often that every time I redo this slide, it's got a new name. But anyway, they give me the permits to collect these animals. So thank you to them. And of course, past and current members of the Smith Lab, a number of which I've mentioned throughout the talk. Funding, NSERC, National Science and Engineering Council of Canada, AXME, the Atlantic Canada Society of Microbial Ecology, no longer around anymore, but they were a great help for funding students in that time, and of course, Acadia University. Thank you very much.